All right. Wow. Okay. Histones, like almost all other proteins, are highly regulated proteins. All kinds of things can be done to histones. Covalent modifications, in other words, attaching things to the histones or to certain parts of certain histones with covalent bonds. All kinds of different things. Each of these things are going to have effects on how tightly the histones bind to the DNA. In other words, it can make nucleosomes tough and strong, or they can make them weak so they can be moved away or even pushed off entirely. And other kinds of changes we still don't know quite how it works in affecting transcription. But histones, now a typical histone, just a little thing, a typical histone forms sort of, part of it forms sort of like a globular structure here, and then you have a little tail on each end. So most of the mass of the histone is this globular, lozenge-shaped structure, but each histone has these kind of tails on one end and another tail on the other end. Generally, these tail regions are involved in interacting with other histones to make the nucleosome and things like that, or with other proteins. But it's especially the tails the tail regions especially get heavily modified. They get heavily regulated. Most of the time when you're regulating histones, you're doing something to those histone tails. They may not attach to other proteins the way they used to, whatever the case is. But there's several things that can happen here. Histones can be phosphorylated at multiple sites. Now those of you who remember cell biology, you all remember cell biology, right? Wasn't it such a great class? Don't answer that. Don't answer that. Or this is being recorded. Okay. All right. Scare all future students up. Well, anyway, okay. Phosphorylation, if you recall, is where you covalently attach phosphate ions to certain amino acids, usually serine and threonine, on target proteins. And that's done by what are called protein kinases, and there are phosphatases, enzymes that remove these phosphates. Now, when you add a phosphate group to a protein, what's going to happen? Phosphates have a double negative charge on them. So, what happens? You add phosphates, which are negatively charged, to highly positively charged proteins. You're canceling these charges out. What that means is that a phosphorylated histone simply won't be able to stick to DNA as tightly because there's not as much positive charge there. So that's one big thing, phosphorylation. In addition, we have acetylation, which also attaches negatively charged acetyl groups. Acetyl groups are acetic acid covalently attached to something. And of course, you would lose the proton here, so we get that. Okay, that's an acetyl group. Once again, you're dealing with negative charges that cancel out the positive charges of the histones. There are other modifications as well that have various effects on the histones and can affect transcription in one way or another. These include things like methylation, where we covalently attach methyl groups, which are hydrophobic to certain amino acids. That can affect the histone interactions with DNA or the histone interactions with other proteins. And there's something called Sumoylation. And Sumoylation, there is a protein that's called sumo, like the wrestler. It's actually a relative of ubiquitin. It's closely related to ubiquitin, which attacks proteins for destruction in proteasomes, but it's not used as a death mark or anything. It has regulatory roles. So here you're covalently attaching to the histones this small protein. for regulatory purpose, and these can do a number of things too. So there's a number of different modifications we can do with the histones that will alter their interaction with DNA or do other kinds of things. Perhaps most important, and there's multiple places happen, are phosphorylation and our acetylation. That's what we're going to focus on here, is the main kinds of things. Okay. Yes? Okay. Attaches sumo. It's all capital letters. And sumo is a protein that's a relative, that's closely related to ubiquitin. But it's a regulatory protein. It's not used to tag proteins for destruction like ubiquitin is. Okay. But it's very similar in sequence. It's small, about 5,000 molecular weight or so. Okay, so that's another type of modification. But let's focus on the phosphorylation and acetylation. All of these histones can be modified by phosphorylation and acetylation in many different locations. Even though histones are fairly small proteins, there's many different phosphorylation and acetylation sites there, and different kinases and phosphatases that do or undo the thing. But basically, if you're going to heavily phosphorylate and heavily acetylate histones just before transcription, lo and behold, what do we see? We see an increase. Okay, we could do this. This would apply to acetylation as well. Okay, we have when we stimulate the cells with hormones, for instance, and when transcription actually takes place. We see certain baseline levels of acetylation or phosphorylation. Within a period after the stimulation, we start seeing the levels of phosphorylation and acetylation go up and up and up, and finally you start getting transcription occurring. And there's a good reason for that. Because once again, if you have critical DNA sequences wrapped around nucleosomes, they're binding so tightly that things like the components of the basal complex, the RNA polymerase, all those kind of things, they can't get to those sequences. So what we've got to do is we've got to get the nucleosomes off of those sequences. We have to somehow get the promoter away from the nucleosome so now the appropriate proteins can bind. You look like you have a question.
What's the first thing for?
Oh, stimulate. In other words, let's say we hit them with hormones, within a short period of time we start seeing increases in phosphorylation and acetylation. But those increases are occurring prior to transcription. Does that help? Okay. All right. So, the idea behind all these modifications, especially acetylation and phosphorylation here, is that now the histones are not binding the DNA as tightly. That's a good thing. So, okay, here's a couple nucleosomes here. Let's suppose right over here, this line represents where a promoter is. We need that promoter. Right now, it's wrapped around a nucleosome. Nothing can get to it. So our first step, we're going to phosphorylate and acetylate the histones. Now, the nucleosome won't stick to the DNA as tightly. The histones won't stick to the DNA as tightly. So that's our first step. Now the second thing is we're still wrapped around, the sequence is still covered up and associated with the nucleosome. We've got to get that nucleosome off of that so it can be exposed so the appropriate proteins involved in transcription can come in. So that's now, we've weakened this thing up, and now we're going to push the nucleosomes out of the way. We're basically going to slide them down the DNA and leave this sequence open and exposed. So we've got to push the nucleosomes down the DNA so that we get the sequences we want exposed so now things can bind to them. So we have to move them out of the way. Okay, now what does that is this. There are these proteins called chromatin remodeling complexes. And what they do is push the nucleosomes down or up, but basically push them away from sequences you need to have exposed. So they're large enzyme complexes. Now, because pushing nucleosomes up or down the DNA takes energy, their attachment to the DNA is weaker. It's not completely abolished. They don't fall off. These chromatin remodeling complexes have to use a source of energy. And the energy they use is ATP hydrolysis. These are ATP-powered machines that act to push. Here, we'll put one here. I'll say CRC for chromatin remodeling complex. Okay, and what we're going to do is hydrolyze ATP. Now, we don't know any structural details about these guys. They're big, they're complex. But we're going to kind of slide this nucleosome down this DNA. And let's do that here. Kind of like so. We slid that nucleosome down the DNA, and lo and behold, our promoter sequence is now exposed. Now other things can bind to it, and transcription can take place. And here again, these chromatin remodeling complexes seem to go into action shortly before transcription. Now, when we're talking about some of the modifications of histones, we see that we have to do things like phosphorylate and acetylate them so that they don't bind to DNA as tightly. But we also have things like acetylases and phosphatases that can remove these things. Remember, when we get a little diagram, there's always a certain baseline you have, even prior to transcription, prior to stimulation. The histones have a certain amount of acetyl and phosphate groups on them already. Now, suppose you remove those. You've gotten rid of some more positive, some more negative charges. Now, those histones will bind even more tightly to the DNA than they did before. So sometimes, if you remove acetyl or phosphate groups, you're going to get those nucleosomes, you're going to get those histones to stick even more tightly to DNA. That means it's going to be harder for that gene to get transcribed. So usually histone acetylation phosphorylation is going to help promote transcription. And frequently the reverse, removing these things, is going to prevent transcription by making them stick even more tightly than they did before. So it's a double-edged sword. These are important ways of regulating these things. They can either weaken the grip on DNA or make it even tighter. And these other things like methylation can sometimes increase transcription, methylation, other places do exactly the opposite. So there's a number of different kinds of things in terms of histone modification. We're realizing more and more, and when we get to what's called epigenetics, which is the idea of stable and frequently long-term and often heritable changes in gene expression. In other words, a change in gene expression that passed from one generation to the next. We're realizing that this epigenetics is a very hot topic in biology now. We know relatively little about it, but it seems to be very, very important. And a lot of that seems to involve modifications to histones, stable modifications that can actually be replicated and effectively repeated onto the new DNA when a cell divides. So we'll look at that a little bit later. But that's going to be a hot field, by the way. Great paper topic. Yes?
So, in order for transcription to occur with the nucleosome, you have to modify the histones by the phosphorylation.
Phosphorylation and acetylation are two big ones. And the reason why is it's adding negative charges to a positively charged protein, so now it doesn't stick to the DNA as tightly. Now we can push those things away. Without that, those chromatin remodeling complexes can't push a nucleosome off, which is sticking too tightly to the DNA, and you can kiss that bye-bye. So I have to weaken its attachment to DNA first before you even have a chance of being able to push them away from sequences. Okay, well, that's a few basic pointers about changes in chromatin. And of course, when you do that, when the DNA is no longer tightly bound to the histones as they were before, now nucleases can get to them and start chopping the DNA up, even the stuff wrapped around the histone core. So that's where you get your DNA's hypersensitive sites. Now it's actually chopping up the DNA that is still wrapped around the histone, but not wrapped as tightly. So nucleases can now get at it and start attacking it. They couldn't before. So that's where we had our hypersensitive sites. You're actually cutting the DNA that's wrapped around the nucleosomes because nucleosomes aren't holding it as tightly. So now they're more accessible to nucleases. Okay, well, and other kinds of things too. Okay, so that's a major idea about changing chromatin structure prior to transcription. Once again, we see more and more the roles of histones in regulating transcription is getting to be more and more than we once realized. At first we thought, okay, it's just organizing, these things are just organizing and packing the DNA so it can fit a lot of DNA into a small volume. And then it looked like, oh, these are general blockers of transcription. Now it looks like the story with histones and histone regulation is much more complex than we initially realized. You can have short-term changes by modifying histones, long-term changes, even heritable changes. And that's probably one reason why histones are so highly conserved, because there's so many different regulatory sites that if you play around and change a single amino acid, you're probably going to screw its regulation up, and God only knows what that's going to do to transcription. It can really do a lot of very nasty things and cause all kinds of problems, failure for development to occur, cell death, the whole 9 yards. Okay, so we've got that. All right, let's see what we do time-wise. Okay, all right.
Dr. Sauterer?
Yes.
I hate to ask this. May I leave just a few minutes early? I have an hour drive and I've heard the road drives at this point.
Okay, yeah. Well, I said at the beginning, I said if anybody for safety reasons feels they should get that. Because I know northern parts are going to get worse. Now I have to drive a little bit west and stuff into Pell City.
I have to go to Talladega.
Okay, all right. It's a little longer trip than me. Yeah. Now, fortunately, I also grew up in the north, so I have had a little bit of experience driving on this. Yeah, as it gets quite dark, the roads will probably get slicker. But apparently it's not going to be a snow deposits on the ground. As a matter of fact, it looks like it's stopped here at this point.
Well, it's on the ground in Talladega County at this point.
Oh, really? Okay. All
Because
right.
it wasn't when I left, I was like, how could it?
Okay, yeah. Yeah, whenever you feel like saying, be careful too.
Okay.
Don't get yourself killed.
Thank
Maybe
you.
pick up the notes from somebody else.
All right. Thank you, Dr. Sauterer. I
Sure.
just want to get up.
Oh, no, I don't blame you. I'm going to face that myself in a couple of hours. But like I say, I'll probably be a little bit better off if say, driven this kind of stuff before.
Right, well,
Growing
not
up.
me, and then I've got
Oh,
something to
sure.
know before.
Yeah, I know. Growing up in Cleveland, Ohio, we do get some experience in that, trust me. We had all kinds of things. All right, take care, good luck on that. Okay,
Thank you.
all right, sure thing.